Welcome back to the deep dive. So today we're tackling a really foundational piece of forensic work, species identification. Before any lab gets to the really high-tech DNA profiling, they have a much simpler question first, right?
Is it human?
Exactly.
Yeah.
Is this stain human blood,
Yeah.
or is it something else entirely? Maybe just ketchup from lunch.
And answering that is absolutely the first critical step in the lab. Our source material, drawing from key forensic biotexts, points out something important. While, yeah, modern labs often rely on DNA quantitation assays now.
Which often check for human DNA anyway, right?
Precisely. They often detect higher primate DNA as part of the process, but, and this is the key thing, those older serological methods we're diving into, they're still incredibly vital.
Okay, vital how? For screening.
For screening, absolutely, for quickly excluding non-human samples, which saves a ton of time and money, and crucially for field testing right there at the scene.
Gotcha. So DNA hasn't completely replaced them. They still have their place.
Definitely.
Yeah.
So today we're really focusing on the mechanics behind those serological techniques. Specifically, the assays built on that amazing specific binding reaction between antigens and antibodies. That's what lets us tell human from non-human.
All right, let's talk tools then. Antibodies. It sounds like engineered biology designed for recognition. Can you break down how these assays actually work? How do scientists see a reaction?
Well, it's fundamentally a lock and key kind of mechanism.
Hmm.
These assays depend on what we call primary binding. That's the first contact, the antibody latching onto its target protein, the antigen.
Okay, lock and key. Simple enough.
But the primary binding itself is usually invisible, so you need a secondary reaction. And the most common one we look for, especially in the classic methods, is precipitation.
Precipitation.
Oof.
Like stuff falling out of solution.
Exactly. When you have the right ratio of antigen to antibody, they link up, forming this large, complex network like a lattice. And this lattice gets so big it's no longer soluble, so it becomes visible as a precipitate, a cloudy spot, or a line.
Okay, makes sense. So how do labs get these specific keys, these anti-human antibodies, to begin with? You can't just buy them off the shelf. Or can you?
Well, you can buy them now, but they have to be made first. It's a standard immunology process, actually. You take purified human serum blood proteins and inject it into a host animal, like a rabbit or a goat, typically.
Ah, so the animal's immune system sees the human proteins as foreign.
Precisely. It mounts an immune response and starts churning out antibodies specifically designed to bind to those human proteins.
And I remember reading that albumin is like the most common protein in serum.
It is, by far. So the resulting antibody mixture, we call it a polyclonal antiserum because it contains many different antibodies targeting different parts of the human proteins, reacts really strongly with human albumin. It's a good general marker for human serum.
Polyclonal, meaning it hits multiple targets on the human proteins.
Yeah, a whole collection of antibodies produced by different immune cells in the host animal. It gives a robust reaction.
Okay, so if that serum antibody mix works well because albumin's so abundant, why bother targeting other things? You mentioned hemoglobin HB and something called Glycophorin A, GPA.
Good question. Because sometimes human fluid isn't specific enough. Hemoglobin, is the protein that carries oxygen inside red blood cells. So targeting HB gives you a much stronger indication you're dealing with blood, not just any human fluid.
Right, more specific to blood itself.
And Glycophorin A, or GPA, takes it another step further. GPA is a protein that's actually embedded in the outer membrane of human red blood cells. It's an even more specific marker just for those cells.
So moving from a general human marker, albumin, to a blood marker, HB, to a really specific human red blood cell marker, GPA. It's about increasing precision.
Exactly. Choosing the right target is crucial.
But just choosing the target isn't enough, right?
Right.
The antibody tool itself needs to be reliable, which brings us to quality control.
Yes, and this is where things get potentially tricky. Remember that lattice formation for precipitation? It absolutely depends on having the right ratio of antigen to antibody. Get that ratio wrong, and the whole test can fail. You can get a false negative.
Whoa, okay, a false negative in forensics? That's bad. Really bad. How did that happen? What are these ratio problems called?
We call them the prozone and postzone effects. Let's break that down. In the prozone, you have way too much antibody compared to the antigen.
Too many keys for the locks.
Kind of. The antibody molecules basically swarm the antigen molecules. Each antigen gets coated with antibodies, but they can't linked up effectively with other antigen-antibody pairs to build that large visible lattice. So
Yeah.
even though the antigen is there, no precipitate forms looks negative.
No. So the test actually fails because you have too much of the reagent designed to detect the target
Okay.
and the post zone.
Post-zone is the flip side. You have a massive excess of antigen, tons of the human protein, but not nearly enough antibody.
Too few.
Right. The few antibody molecules bind antigen, but there's so much free antigen floating around that, again, you don't get efficient cross-linking to form that visible precipitate. Same outcome, false
Right.
negative. Missing that distinction can, well, derail an investigation.
Okay, prozone, post zone, both bad. How do labs make sure they're in that sweet spot, that Goldilocks zone, with the right ratio?
Through a quality control process called titration. It's fundamental. Basically, you take your antibody solution, your anti-serum, and you make a series of dilutions, progressively weaker solutions.
Like 1 to 10, 1 to 100, 1 to 1,000, that kind of thing.
Exactly. And you test each dilution against a standard known amount of the antigen. You're looking for the weakest dilution that still gives you a clear positive reaction.
And that weakest dilution tells us something important.
It tells you the titer. The titer is just the reciprocal of that highest dilution that worked. So if the 1 to 1,000 dilution worked, but 1 to 2,000 didn't, the titer is 1,000. It essentially tells you the strength or the amount of functional antibody you have. It lets you calculate the right concentration to use in your actual tests to avoid prozone and postzone.
Okay, so titration handles the quantity problem. What about specificity? Making sure the antibody only reacts with human stuff. You mentioned cross-reactivity earlier.
Right, specificity is critical. Now, most anti-human antibodies will show some cross-reactivity with higher primates, chimps, gorillas. Our proteins are pretty similar.
Which, forensically speaking, isn't usually the biggest worry, I guess. Unless the suspect is an escaped gorilla.
Yeah, usually not the primary concern. The critical check during validation is making absolutely sure the antibody doesn't react with common domestic animals. Dogs, cats, cows, pigs, rodents. Stuff you might actually find at a crime scene. If your anti-human antibody lights up for dog blood, it's useless. Worse than useless, actually.
Yeah, misleading. Okay. Anything else affecting these reactions? Environment.
Oh, definitely. These are biochemical reactions, so conditions matter. Things like the salt concentration. The ionic strength of the buffer solution can affect binding. Usually higher salt inhibits it.
Inhibits, so it makes it harder for the antibody and antigen to connect.
Correct. On the flip side, labs sometimes add things like polyethylene glycol, PEG. It's a large polymer that effectively decreases the solubility of the proteins in the solution.
Making them more likely to precipitate out.
Exactly. It can help facilitate that lattice formation, make the reaction easier to see. And of course, temperature and pH have optimal ranges too. You need to control the environment.
Okay, let's shift gears to the modern stuff. The things investigators might actually use out in the field. You mentioned immunochromatographic assays, like test strips.
Exactly. Think of like a home pregnancy test strip. Same basic technology. They're fantastic tools, rapid, generally very specific, sensitive, and portable. Perfect for field use or quick lab screening.
And they work differently from the precipitation tests, right? You mentioned a sandwich.
They do. They typically use what's called a labeled antigen-antibody sandwich method, and this is where monoclonal antibodies often come in.
Ah, okay. We talked about polyclonal antiserum before, the mix of antibodies. Monoclonal is different.
Very different. Monoclonal antibodies are engineered to recognize just one specific site, one epitope on the target antigen. They're much more uniform and consistent than a polyclonal mix. So these strips often use monoclonals for better control.
Got it. So let's look at the common types. First, the ones targeting hemoglobin, HB, like the ABA card hemotrace or hexagon OBTI tests. How does that sandwich work?
Okay, so picture the strip. In the sample well area, where you add the extract from the stain, there are labeled monoclonal anti-HB antibodies, usually labeled with something visible, like tiny red particles.
Labeled keys.
Right. If human HB is present in the sample, it binds to these labeled antibodies. Then the liquid flows up the strip by capillary action, carrying this labeled antibody HB complex along.
Okay, it's migrating.
It migrates until it hits the test zone, usually marked T. Immobilized there, stuck to the strip, is another antibody against HB, often a polyclonal one, or a different monoclonal targeting a different part of the HB molecule.
Ah, the other piece of bread for the sandwich.
Precisely. This immobilized antibody captures the migrating complex. So you get immobilized antibody, HB antigen labeled antibody. a sandwich. And because the labeled antibody has those visible particles, you see a pink or red line form at the T-zone.
Makes sense. And there's usually a control line, too.
Absolutely essential. Further up the strip is the control zone, marked C. This zone typically has immobilized antibodies that capture any labeled antibody, whether it's bound to HB or not. So a line must appear at C just to show the liquid flowed correctly through the strip and the reagents were working. No C line means the test is invalid, regardless of the T line.
Okay. 2 lines, T and C, for a positive, C line only for a valid negative. How sensitive are these HB tests?
They're very sensitive, much more sensitive than older presumptive tests like Kastle-Meyer. They can detect HB down to around 0.07 micrograms per milliliter, really tiny amounts.
But you hinted at a catch earlier.
Yeah. Their high sensitivity can be a double-edged sword because hemoglobin, or at least traces of it, can sometimes be found in other human body fluids besides blood, things like semen, saliva, even some urine samples.
So a positive result on an HB test doesn't absolutely guarantee it's blood, just that it's likely a human fluid containing some HB.
Correct. It's a limitation in specificity. Plus, they can suffer from that high-dose hook effect, the postosome problem we talked about.
Yeah.
If you put a drop of pure undiluted blood on there, sometimes the antigen excess is so extreme you can get a false negative.
Okay, limitations noted. Which leads us to the other target, Glycophorin A, GPA. You said assays like RSID blood targeting GPA were a big step forward.
A huge step forward, mainly addressing those limitations.
Why? What makes GPA so much better as a target?
Mm.
This seems like the key takeaway for listeners here.
It really comes down to specificity and just overall reliability. Remember, GPA is stuck right there in the membrane of human red blood cells. It's not typically floating around in other body fluids.
Okay, so inherently more blood specific.
Exactly, and validation studies confirmed this. The GPA assays showed essentially no cross-reactivity with other human fluids like semen, saliva, urine, breast milk. None of that stuff. If you add a positive on a GPA test, you have much higher confidence it's actually human blood.
What about cross-reactivity with animals?
Also excellent, they showed no cross-reactivity even with non-human primates, let alone common domestic animals. So highly human-specific and blood-specific.
And the hook effect.
That was another major improvement. These GPA assays were specifically designed and validated to avoid the high-dose hook effect. You can test pretty concentrated blood samples without worrying about that particular type of false negative. Plus, they are also highly sensitive, detecting down to maybe 100 nanoliters of blood.
Wow. Okay, so better specificity across the board and less prone to the hook effect. That sounds like a clear winner. All right, let's step back in time a bit now and unpack some of those classic methods you mentioned earlier, the ones relying purely on visible precipitation in tubes or gels.
Sure, these are the foundational techniques, really important to understand the principles. The simplest is probably the Ring Assay or Ring test.
How'd that work? Sounds straightforward.
It is. It's a form of double immunodiffusion, but done vertically in a small test tube. You carefully pipette the antibody solution, the anti-serum, into the bottom of the tube. It's usually denser. Then you gently layer the sample extract from the suspected blood stain on top.
Layering them without mixing.
Exactly. Because they have different densities, you can create a sharp interface between the two liquids. Then you just wait. If the sample contains the human antigen, it diffuses downwards, and the antibody diffuses upwards into that interface zone.
And if they meet in the right ratio.
You get that precipitation reaction we talked about, forming a visible white ring right there at the interface between the two layers. Usually takes several minutes to maybe half an hour. Simple, elegant, visual confirmation.
Okay, the Ring test. What came next? Gel-based tests.
Right. Moving the reaction into a semi-solid medium like an agarose gel gave more control and information. The classic example is the Ouchterlony assay, or double immunodiffusion in gel.
Ouchterlony, how does that work?
You prepare a flat layer of agarose gel, maybe in a petri dish. Then you punch small wells into the gel in a specific pattern, say a central well for the antibody, and surrounding wells for your known human blood control and your unknown samples.
So everything diffuses out from the wells into the gel.
Correct. The antigens from the samples and the antibodies from the central well diffuse outwards, towards each other. Where they meet in optimal proportions, they form a line of precipitate in the gel between the wells.
Okay, so you'd see lines forming.
Huh.
But you said this gives more information than just yes-no.
It does, because you can compare the precipitate lines formed by different antigens against the same antibody. There are 3 key patterns you look for telling you about how related the antigens are.
3 patterns. Okay, what are they?
First is identity. This is what you want for a species test. If the line formed by your unknown sample completely fuses, like merges smoothly, with the line formed by your known human control, it means the antigens are immunologically identical. Same stuff.
Makes sense. Fusion means identity. What else?
Second is non-identity. If the two precipitate lines completely cross each other, forming an X, it means the antigens in the two wells are totally unrelated. The antibody is reacting independently with each one.
Okay, crossing means unrelated. And the third?
The third is partial identity. This is really interesting. Here the two lines merge, but one of the lines continues on past the junction, forming a little spur.
A spur. What does that tell you?
The spur indicates that the antigens share some common features, some reactive sites or epitopes, but one of them, the one forming the spur, has additional unique epitopes that the other lacks. They're related, but not identical.
So, for forensic species ID, if you saw a spur between your sample and the human control,
That would not be a positive ID for human. It might mean it's from a related species, maybe a primate sharing some markers, but critically, it's not identical. For a positive confirmation, you need that smooth fusion, that complete identity, no spur.
fascinating level of detail from just watching lines in a gel. Okay, one more classic technique, crossed over electrophoresis. Sounds like a hybrid approach.
It is. It combines immunodiffusion in a gel, like Ouchterlony, with electrophoresis using an electric current to speed things up.
How does adding electricity help?
It dramatically speeds up the movement of the antigen and antibody towards each other. Instead of just relying on slow diffusion, you use the electric field. Typically, you'd place the anti-human antibody well near the positive pole, the anode, and the sample wells near the negative pole, the cathode.
Why that way round?
Because most proteins, including antibodies and antigens, carry a net electrical charge depending on the buffer pH. Under typical conditions, they'll migrate in the electric field. You set up so the antigen and antibody migrate towards each other
So they meet much faster in the middle.
much faster. Instead of hours or days for diffusion, you can get a result, a sharp precipitate band forming between the wells in maybe 30 minutes to an hour.
That's a big time saving.
Yeah.
But adding electricity, does that introduce new problems?
It can. You still have the risk of false negatives from things like the post-zone effect, just like the other precipitation methods. But now you've added electrical variables. You could potentially put the wells in the wrong place relative to the poles, or run the current in the wrong direction. Yeah, big oops. Or use too high a voltage or current. That can generate heat, which could potentially denature the proteins, cook the antibodies or antigens, and prevent them from reacting properly. Or using the wrong buffer could mess up the migration. So faster, yes, but also more ways for things to potentially go wrong if you're not careful. Hashtag #TakeTagOutro.
Okay, wow. We've covered a lot, from basic antibody reactions to modern test strips. So let's try to synthesize this. What's the big picture takeaway for someone listening?
Well, I think the first thing is just reinforcing that species identification is that absolute baseline check in forensic biology. Got to know what you're dealing with.
Right. And even though DNA quantitation often handles this now,
Those serological methods, both classic and modern, are still indispensable for rapid screening, field tests, and honestly for understanding the fundamental biochemistry of how we even do biological identification.
and within those methods, we hit some key points. Watching out for prozone and postzone effects.
Absolutely critical. Understanding titration and titer is key to antibody quality control and avoiding those dangerous false negatives.
And realizing the leap forward that came with the newer immunochromatographic strips, especially those targeting GPA.
Definitely. The move to Glycophorin A assays solved major headaches with the older hemoglobin tests, better specificity against other body fluids, less worry about the high-dose hook effect. It's a much more reliable indicator of actual human blood.
It really is amazing seeing the evolution from literally watching for a cloudy ring in a test tube to getting a specific pink line on a strip in minutes out in the field.
It is. And that brings us, I think, to a good final thought for you to consider. We have these incredibly sensitive, sophisticated tools now, but
There's always a but.
there's always a but. The complexity, even in seemingly simple test factors like prozone, postzone, potential cross-reactivity, how degraded the sample might be, the specific target molecule chosen, HB versus GPA, it all highlights that interpreting a result isn't just about seeing a line or a ring.
It requires critical thinking.
Constant critical evaluation. You always have to ask yourself, what are the limitations of this specific test under these conditions with this sample? What else could possibly explain this result or lack of result? It's that critical overlay, that constant questioning that elevates using a technique into practicing real science. You always have to wonder, what else might be going on here?